Okay, so we're going to begin. So just a little comment. The time restriction is 3 minutes, not 4 minutes, as was mistakenly. So it's 3 minutes, and it's strict 3 minutes, and the audience is very instrumental in making sure that it's 3 minutes. So in 3 minutes, you're going to hear some bells and rings, and so it's very important to everybody to let the speaker know that it's time to step down. Okay? So 3 minutes, and under no circumstances any extra time. Other than that, 3 iPads, 3 prizes, no pressure, and may the best women win.
Okay. Also speak to the mic. Okay? So let's start. Amir Endelman. Amir Endelman. We're starting the time. He's not here. Okay, very good. We're doing excellent in time. Okay. Next. Anna Skomparyn.
So, good evening. This study is a side-by-side comparison of 2 actively targeted delivery systems for anti-cancer drug. The first system is a polymeric prodrug based on a pullulant backbone modified with folic acid as a targeting agent and doxorubicin as anti-cancer drug. The second system is a commercially available pegylated liposomal formulation of doxorubicin, known as Doxil, and we modify it with folic acid in order to confer active targeted properties to the system as well. So we first investigated the internalization and the intracellular trafficking of doxorubicin and of its drug delivery systems. Here we see confocal images of KB cells that are known to overexpress the folate receptor. KB cells treated with doxorubicin and with the polymeric prodrug of doxorubicin with and without the targeting agent, and of the liposomal formulation with and without the targeting agent. As we can see, in 4 hours, both the polymeric system and the liposomal formulation modified with the targeting agent shows internalization of doxorubicin and colocalization in the nucleus. This is reflected in higher anti-cancer activity, as we can see in proliferation assay, where the polymeric prodrug of doxorubicin and the liposomal formulation modified with folic acid shows higher anti-cancer activity compared to the 2 formulation without the targeting agent. We finally investigated the in vivo ability of reduce the cardiotoxicity of doxorubicin, as the cardiotoxicity is one of the main issues in the clinical use of the drug. And we perform ultrasound echocardiography on the heart of the mice treated with doxorubicin and with drug delivery systems, and we can see that both the liposomal formulation and the polymeric formulation are able to maintain the left ventricle systolic volume and the ejection fraction in a physiological level, as opposite as doxorubicin. Those results were confirmed also by histological analysis of the heart of the animals treated with doxorubicin and with its drug delivery system. In fact, we can see that the animal treated with liposomal formulation and with the pegylated prodrug, with the polymeric prodrug, showed lower left ventricle volume and thicker wall of the left ventricle. In conclusion, we synthesize and characterize 2 folate receptor targeted drug delivery system of doxorubicin. Folic acid indeed promotes internalization in folate receptor overexpressing cells, and the drug delivery system maintained.
Thank you. Next speaker. Ela Markowski.
Shalom. I'm going to present my work on polymer therapeutics designed for combination therapy for breast and ovarian cancer. This work was done in collaboration with Chaim Cohen in the lab of Ronit Sachi-Fainaro in the Faculty of Medicine. So in order to grow, a tumor needs to build new blood vessels. However, as we can see in these videos, the blood vessels are not normal. They're enlarged and they're not organized, and most importantly for us, nano-sized macromolecules such as polymers can leak out of this tumor blood vessels, but can't leak out of normal blood vessels. We can exploit this property of tumor blood vessels to target drugs to tumors. This by conjugating small chemotherapy drugs to a nano-sized polymer, we can direct them specifically to a tumor and therefore reduce the toxicity of these drugs because they will be mainly accumulated in the tumor and not in healthy tissues. In addition, this polymer conjugate is an ideal platform for combination therapy because we can conjugate two different synergistic drugs to the same polymer chain. So the two drugs we chose to work with are paclitaxel and doxorubicin. Both of these drugs are widely used in the clinic and they have severe side effects. We also previously examined the anti-cancer activity of the combination of these drugs on cancer cells, and we found they have a synergistic activity on several cancer cell lines, including breast and the ovarian cells. We therefore conjugated both of these drugs to a polyglutamic acid and created the paclitaxel PGA, paclitaxel doxorubicin conjugate, and examined its activity in vitro and in vivo. You can see here the anti-proliferative effect of this conjugate, marked here in red. We mainly wanted to see that the drugs were successfully conjugated and they retained their anti-cancer activity and released in their active form. We also examined this internalization and the release of doxorubicin, which is a fluorescent molecule by confocal microscopy. You can see that by 24 hours this conjugate is internalized and the doxorubicin is released and is accumulated in the nucleus, which is its target site. And next we examined the activity of the conjugate on tumor-bearing mice with breast cancer. You can see the conjugate again here in red. It had the highest anti-tumor activity. It maintained the lowest tumor volume of the mice of all the other groups compared to a combination of free drugs and the drugs conjugated to each polymer chain separately. It also didn't have any effect on the weight of the mice. They gained the weight as they were supposed to, as opposed to free drugs, which in these groups bestow a significant weight reduction of the mice, which resulted in longer survival. Thank you.
Thank you. Okay, Edith Velis.
Okay, hello. Okay, so this work was done with all these people which helped. Okay, and the work objective is nanodoping proteins and exploring the dope protein self-assembled monolayer properties. Now, when I say doping, I don't mean in the conventional way. Okay, we, in a controlled way, bind small molecules to the protein molecules in hope to change the protein properties, okay, and we would like to do that, okay, because we would like to explore charge transport mechanisms in the biomolecules, and also we would like to gain doping at the nanoscale. But when you work with proteins in solid state, okay, there is a big challenge, and the challenge is due to protein surface interactions, which cause the protein molecules to undergo conformational changes. And in simple terms, as you can see in the image here, okay, the protein molecules actually unfold, okay, and lose their native structure. So we asked ourselves two new questions. Okay, does the doping affect these surface-induced conformational changes in the protein molecules, and how do these changes, okay, affect the self-assembled monolayer properties? So these are the experimental surface characterization methods that we used in order to analyze our SEMs, and the surface coverage simulation that we used. And I will just, go back, and I will just show you what is I of it. Okay, what I think is the most intriguing result, okay, and which is that we in fact found a connection between doping the protein molecules and the structure of the molecules in the SEMs, okay, and what you can see here is actually that the doped molecules retain a higher degree of their original structure in solution, and it's actually 2 times more, okay, of its original structure. So if you want to hear more about it, I'm poster number 5, okay, and I'll be waiting to talk to you, so good luck for everybody.
מיכל שבח.
Hello, my name is Michal Shavach, and today I'm going to present to you a work about nanocomposites made of fibers and gold nanoparticles for cardiac tissue engineering. So myocardial infarction, or a heart attack, is caused when one or more blood vessels that lead blood to the heart itself are suddenly blocked. This causes aerial cell death and can lead to deterioration in the heart function, which may cause a heart failure and eventually the death of the patient. This is not my error.
Okay. Go back.
So the aim of tissue engineering is actually to regenerate the damaged tissues or organs. The basic principles of tissue engineering is to combine cells with a scaffold that's going to mimic the extracellular matrix of the organ. It was previously found in our lab that incorporating gold nanowires into a spongy scaffold can elevate the conductivity of the scaffold. Conductivity of the scaffold is very important because most of the materials these scaffolds are made from are initially not conductive. But on the other hand, the conductivity is crucial for proper cardiac function. The drawback of using these scaffolds was that they were sponges. They did not mimic the fibers in the natural extracellular matrix of the heart. So what we have done in my work is to generate a scaffold made of fibers using electrospinning method. Then we introduced gold nanoparticles by simply evaporating gold onto these fibers with different nominal thickness. So we first examined the nanoparticles on these fibers, and we found that they were about 250 nanometers in diameter, similar to the fibers in the natural cardiac matrix. Then we seeded cells on these scaffolds, and we found that when cells were cultivated on scaffolds without the gold nanoparticles, on the left, they remained round and they did not assemble into a functioning tissue. But when we raised cells on the scaffolds with the gold nanoparticles, like on the right, they were elongated and aligned and assembled into a functional tissue. When we wanted to see the function of these scaffolds, as can be seen in this movie, we saw that the scaffolds with the gold nanoparticles are much superior to the scaffolds without the gold nanoparticles regarding to contractility rate and contractility amplitude. I would like to thank Marianne Gertner Fellowship for this work and my lab students. Thank you.
Hello everyone, I'm Eitan. I'm from Ran Perelson's lab in the medical school. So our lab is interested in molecular mechanisms of neuronal communication, and specifically in motor neurons. These neurons are special because they have formed the longest cell in the body, emanating from the spinal cord of the vertebrate and into the target muscle, and we mainly know their role in activating the muscle via neuronal signaling, but also they receive important signals from their targeted tissue in the form of secreted proteins, which are known as neurotransmitters. One of these is the BDNF, the brain-derived neurotrophic factor, which is picked up by specific cell surface receptors on the neuronal surface. And what is interesting about this neurotrophin, the BDNF, is that it activates two kinds of receptors. One is its main receptor, the TrkB, and another one is the P75, which is a common receptor for several neurotrophins. And when these two receptors work together, we know that they activate with high efficiency the canonical pathway, which promotes survival, growth, and axonal elongation and synapse maintenance of BDNF, while when P75 works independently of TrkB with other co-receptors, it can mediate completely opposite signaling events. And what we're interested in is how these two receptors are organized along the cell surface, because our idea is, and this was not fully shown, that they can form alternative signaling complexes on the surface, and that these two alternative signaling complexes mediate the alternative signaling outcomes, and finally determine the fate of neurons, either to survival or to death. And this is something we're interested in. And so to tackle these questions, we have established a live imaging-based system using mainly turf microscopy in living neurons. And so we are using this imaging-based assays to follow receptor particles along the cell surface via spot detection and particle tracking. We follow their motility and diffusion via fluorescent recovery after photobleaching. And we're also interested in how these receptors are aggregate with the other membranal components, such as lipid macrodomains. And furthermore, I hope you can see that, is that we're following also microtubule dynamics near the cell surface. We're specifically interested in this component of the cytoskeleton because we believe it has a role in interacting with the membrane and with the membranal receptors and determine their localization and accumulation into clusters. And so I hope that piqued your interest. This is the title of my poster. It's number 8. I hope to see you there.
Sharon?
Hi, my name is Sharon Fleischer, and I will continue Michal's talk about cardiac tissue engineering. And I will talk about engineering the cardiac microenvironment. So, in vivo, the cells are surrounded by the extracellular matrix, which is a natural web of organized fibers. What you see here is the cardiac ECM. Each tissue has a unique architecture of the ECM. And this architecture of the cardiac ECM provides topographical cues for cardiac cells that control their behavior and organization. Our goal was to fabricate a structurally biomimetic scaffold. And our hypothesis was that when seeding cardiac cells on a scaffold which provides them the cues they see in vivo, then they will reorganize and assemble into a functioning tissue. So our first step was to investigate the architecture of the cardiac ECM. And we made several key discoveries. One, we saw that it is composed of microscale fibers and nanoscale fibers. Second, we saw that it is composed of aligned and randomly oriented fibers. And the most interesting discovery is we found a unique population of coiled fibers which are responsible for the contraction and relaxation of the heart muscle. So our next step was to engineer a scaffold that mimics these features. And as you see here, we were able to control fiber size, and we fabricated microscale fibers and nanoscale fibers. Then we controlled their alignment. And the hardest thing we did, as you see here, we were able to fabricate coiled fibers as we saw in the heart. Next, we sought to investigate the effect of these biomimetic scaffolds on cardiac cells. And when we seeded cardiac cells on these scaffolds, as you can see here, they gained a natural morphology and gained cell-cell interactions as seen in vivo. And most of you will have to trust me on this. And the most important thing is that we see that the cells reorganized and assembled into a functioning and spontaneously contracting cardiac tissue. These results indicate that structurally biomimetic scaffolds have a great potential in cardiac tissue engineering. And our next step is to try to improve heart function after myocardial infarction using these biomimetic scaffolds. Thank you.
Good afternoon. My name is Alex, and I'm from Professor Fernando Potolsky group. If you want to discover novel methods for preparation nano building blocks, I highly recommend it to attend our poster. In our poster, you will find a method for preparation of super flexible semiconductor nanoribbons. This is our poster. Look at it, remember, and don't miss it. In the poster, you can find a method for preparation of nanoribbons. Shortly, it's based on synthesis of core shell nanostructures, top curving of the shell part, and etching of the core part. Our nanoribbons, our method allows us tight control over physical and chemical properties of nanoribbons. We have ability to control the size, the shape, the doping of nanoribbons, etc. They can be used as building blocks for electronical devices due to their properties, flexible properties, as can be seen down. They are promising building blocks for flexible electronics or 3D electronics. Additionally, in the poster, you can find the explanation for unwrapping mechanism, which is not so trivial. So, thank you a lot. Come to our poster and don't forget to vote us.
Tai, Barnea, Nir Turko, presented by Nir.
Hi, I'm Nir. I will present 3 of the projects that are done under the supervision of Nati Shaked. The first is Itai Barnea's project. Essentially, when we want to look at biological cells which are transparent samples, there's a problem with bright field light microscopy because there's only one beam that goes through the sample, and it gives us poor contrast, as you can see in the upper image. It gives us poor contrast and only a 2-dimensional information of the sample. One of the solutions to this problem is interferometric phase microscopy, which is the introduction of another arm, a reference arm, which does not go through the sample, and it interferes with the sample with the sample beam upon the camera, and from that interference we can get the optical path difference between these 2 beams. Itai used the tau interferometer, which is a technique that was developed in our lab, and I will present it in about 6 minutes. The main thing about the tau interferometer is that it allows us the acquisition of phase images with resolution of sub-nanometer resolution. Itai used this technique to investigate the elasticity or the fluctuations of various cells. You can see here the upper row represents the primary, and the lower row represents the metastatic lesions of the same of the cells from the same person. And his hypothesis is that the metastatic lesions, which are supposed to travel through the body, are supposed to be more elastic, so they will fluctuate more. And as you can see in the fluctuations map, this is the standard deviation of about 200 consecutive images, and the standard deviation of the metastatic cells is higher.
Zohar Tzafrir.
Hi, my name is Zohar. I'm from Tamir Tuller's lab. Don't see it in the slide, but I'm going to talk to you, I'm going to present a poster about my current research, which is the study of splicing and the mechanism of splicing regulation. Splicing is a process in which pre-mRNA transcripts are being maturated, and where introns, which are intervening non-coding region inside the transcript, are being spliced out, are being removed, leaving only the retained coding sequences called exons. Splicing is in eukaryotic cells is a crucial regulatory stage or step in the gene expression, in determining the gene expression, and is also allows us to make more than one isoform out of a single gene, what is called alternative splicing. Splicing is done by the spliceosome, which is a complex, a very large complex of proteins. The spliceosome recognizes the intron boundaries based on those consensus sequences in the 5' and 3', and every gene has its own characteristics or properties of splicing efficiency. We also know that the RNA secondary structure, that means the way the mRNA folds, is important and affects splicing. For example, by masking or exposing those sequences, which can affect the way the machinery of the spliceosome recognize those sequences. We are using, you don't see it, but we are using algorithm, computer algorithm, in order to calculate or predict the RNA fold and calculate the folding energy. In general, I can say that the lower the energy, the stronger the folding is. The higher the energy, the weaker the folding is. And we are asking some questions about it. For example, does pre-mRNA folding affect splicing efficiency in a systematic manner? Does it affect intronic evolution? Can we model those effects? And can we design intron to our needs? Here are some teasing results from our research. If you are interested and you want to find and discuss about it more, and get answers for at least some of those questions, you can come to poster number 12. Thank you very much.
Neil again.
Okay, as promised, I now introduce Pinchas Gershowitz and Nati Shaked's tau interferometer, which is an add-on on a regular light microscope. It's currently under development. You can see it's pretty small. It has a small number of optical elements. It's compact, it's portable. We have 3 of those in our lab, and all of them work fine because it is very easy to align. Anyone who worked with the optic interferometer can build this one. It's a low-cost interferometer. It's easy to use for the regular user. We only expect the user to place a phase or a cell sample upon the microscope, and he instantly gets the sample phase. The most important thing about it is that they achieved to lower the noise sensitivity, to lower the noise, to get a better sensitivity. And they done so by introducing the beam splitter after the light passed through the sample. This ensures a smaller amount of smaller distance, which is different between the sample and the reference arms. You can learn more about it in the poster number 13, I think. These are some of the results. You can see on the left the tau interferometer imaging of writing omni group, and on the right, normal Mach-Zehnder results of the same sample. I guarantee that he did his best on the right results also. And this is a more complicated sample. And that's it. See you again in a few minutes.
Orberger?
Hi, my name is Or, and I'm a student in the lab of Professor Ehud Gazit, and I would like to present my work on the self-assembly of PNA, or short peptide nucleic acids. So molecular self-assembly is the process by which simple molecules, which we can be related as building blocks, they arrange into more complex and ordered structures. So in biological systems, we have two main groups of building blocks. We have the amino acids, which compose all the peptides and the proteins, and on the other hand, the nuclear bases, which DNA and RNA are made of. And in the recent decades, chemists were able to synthesize new hybrid building blocks. And one sample of such hybrid building blocks is PNA. And PNA has many advantages over DNA or RNA. It can actually bind DNA or RNA with higher specificity and affinity than that of two strands of DNA. And it has very high stability, both thermal and biological. It can't be digested by any enzyme. So in my project, I synthesize short peptide nucleic acids molecules, and I try to self-assemble them into other structures. And here you can see a few samples which I was able to form, and I would love to tell you much more about them near my poster. Thank you very much.
Ron Feiner?
Hi, I'll talk to you again about cardiac tissue engineering. You've already seen this slide. As Sharon told you, when a vessel supplying oxygen and blood to the heart gets blocked, you've got a myocardial infarction, and you need to replace a piece of the heart. And that's what we try to do in our lab. We try to generate replacement tissue to treat the heart. What I'm trying to do is a bit different. We're trying to develop a device on which we can regenerate a tissue and also monitor it and control it. What we've created is a small device made of SU8 and gold electrodes interspersed inside, and we've been able to seed cardiac cells on it by first we use biomaterials on the device, and then we see the cardiac cells, so they have their natural environment, like the extracellular matrix in the heart. And then we can connect it to the computer and monitor it, as well as stimulate it or try to release substances like drugs or growth factors into the cardiac tissue. We're also trying to make this device as small as possible, and it's supposed to be flexible and freestanding as well as robust, so we can withstand the contractions of the heart. We want to release drugs, monitor pH and heat and mechanical sensing, and also integrate it with piezoelectrical fibers so the cardiac cells themselves can supply the electricity to the device. Meanwhile, we've created the device out of SU8 and gold, and we've used electrospinning to put on fibers on it and seed cells. And we've been able to connect it to the computer and monitor, and as you can see, some action potentials generated from the cardiac tissue that we've seeded and cultivated on the device. And if you want to hear more about it, I'm poster 19.
Hi, my name is Michal. I'm from Yoram Dagan's group, and I'm presenting a poster on the evolution of the Fermi surface of copper dot bismuth selenide with carrier concentration. An electron in a metal can have its spin oriented in some random direction. A topological insulator is a special system where the charge carriers, which can be either electrons or holes, have their spin pointed perpendicular to their direction of propagation, and they can only propagate on the surface of the material. And this is special because you can have less backscattering is suppressed, and you have better transport. A topological superconductor is something even more exotic, where you can find some exotic particles called Majorana fermions in the cores of vortices on the surface of the material. Let's move. Okay. So what we studied copper-doped bismuth selenide with different doping levels with the two tools of ARPES, angular resolved photoemission spectroscopy, and Shubnikov de Haas oscillations, which we measure in high magnetic fields of up to 33 Tesla. And what we find, we were able to map the Fermi surface of these materials in the different doping levels. And we find that at the doping level where the system is actually superconducting, it doesn't qualify as a topological superconductor, and it actually probably can't hold Majorana fermions on its surface. And if you want to hear more about it and see some cool results, come to poster number 20. Thank you.
Nir Turko, I believe. This one. Nir Turko.
This is my last time, but the most important one, because this is my work. What I did on this project was to image nanoparticles that were well below the diffraction limit towards photothermal super resolution. So here's our nanoparticle. It's about 60 nanometers in width, and we want to image it. So we try to look at it with an ordinary light microscope, and we see nothing because it's below the diffraction limit. So we add the quantitative phase microscopy, which we saw earlier, and we still see nothing. Still because it's below the diffraction limit. But if we incorporate a photothermal arm, we can see with high contrast the center of the nanoparticle. How does it work? Essentially, when illuminating the nanoparticle with the right excitation beam at the right frequency of 532 wavelength of 532 nanometers, we will get what is called Plasmon Resonance. This Plasmon Resonance actually hits the area and causes phase fluctuations. So if I excite the nanoparticle with a frequency of 30 Hertz, I will get a signal at 30 Hertz. The signal is the phase fluctuations in time. Here we see the results of a single pixel. If we look at all of the pixels of the image, we can see the beautiful nanoparticle which you saw earlier, and the amplitude of the Fourier signal which we see here actually rises as we get closer to the nanoparticle center. We can use it to conjugate it to cells, to biological cells. Here you see on the left the confocal images of a conjugated cell, and this image was it's in high resolution, but it took about 1 minute to acquire. While my system is able to acquire about the same image in less than a few seconds and in video rate. We can see about a 75 to 80% match between the methods, but my method is able to dynamically image the photothermal map of the sample. If you want to know more, you can come. I'll be in 11, 13, and 22.
Evgeny.
Hello everybody, my name is Evgeny. I'm going to present you about modeling the effect of adsorbed polyelectrolytes on charged surfaces. And I emphasize the word charged because it's something that wasn't done yet. This work was done with the supervision of Dylan Goldinsky and Dr. Aviv Natan. And let's go. So why do we do it? Because in recent years we see a growing interest in depositing combined layers of nanoparticles covered with polyelectrolytes, which are both a physical structure and a functional medium. How do we deposit or do these films? Usually, or more promising method is actually electrophoretic deposition, and we were interested in why this is here, why bare particles, namely without polyelectrolytes, deposit in a rough manner rather than a smooth manner, which can be seen with soft particles, and why the deposition of soft particles is slower, where in theory it should be faster. So actually we went back to classical models. In 1910 was the Gui, Chapman, and Stern layer, Stern models for charged surfaces, namely hard surfaces. And in 1962 it was soft surfaces, and mostly in a linear scale. In 2013 we model this kind of surface, namely charged surface covered with polyelectrolyte. With some mathematical abracadabra, we managed to make this plot, namely a full potential profile of charged surface covered with polyelectrolyte. And we see, if I have a laser here, never mind, we see that the blue line is the original Gui-Chapman model, and we see that the polyelectrolyte is actually a screening medium, better screening medium than the solution in any case. And plus there are a lot of interesting results regarding the charged surface and polyelectrolyte brush interface in aspects of charge concentration and potential. And regarding electrophoretic deposition, we stop here.
Thank you. Gilad Cohen.
Hi, my name is Gilad. I'm a student in Professor Hanin's group. Okay. For hundreds of years, researchers try to understand how the brain works. So far, they know how individual neurons communicate, but communication in a modular network, such as in the brain, is still an enigma. So, my goal in the research is to understand how the topology of the network affects its activity. So in the lab, we utilize two clusters. Here you can see cluster A and cluster B. Each cluster is a bunch of neurons. It's a lot of neurons with high connectivity. And this bundle is very little connectivity between the clusters. We simulate the topology, and we see that new network burst in cluster B can propagate to cluster A, and they can stay in cluster B without a transfer to cluster A. We see that as we increase the connectivity ratio, in other words, as we make the neurite bundle thicker and thicker, then the clusters become more synchronized. Also, by canceling inhibition, we see that the clusters become more synchronized. And we also record in the lab the clusters using a multi-electrode array, and we see that if the higher the activity in one cluster, the higher the probability it will transfer to the other cluster. And for the future, we want to demonstrate learning and memory imprinting. To do so, we propose a real-time controller that will stimulate the network based on past activity. That's it.
Okay, Vital?
Hi. My poster presents a method of sensing nanoscale displacements. So what is it good for first? Many kinds of sensors, such as accelerometers and gyros and pressure sensors, they work with the displacement sensing. And in many examples, these sensors can be life-saving, and we want to gain high precision, and for that we need high resolution of sensing. So, in order to explain the principle of the MEMS that I work with, I want to take you back to the Inquisition era, in which it was pretty much about chasing and torturing heretics of the Catholic Church. And here we see an example of torture that stretches the human body to death. So don't worry, our device is not meant for human, it's meant for nanostructures. And we want to present the device. It consists of a curved beam, right here, that is anchored in this end, and that in this end it's connected to a side beam, which is relatively free to move. And on this device we grow by CVD carbon nanotubes in a localized manner, bridging between the side beam and this electrode. So how does it work? The curved beam is electrically grounded, and we apply positive voltage on this electrode. It's about tens of volts. So it pulls the curved beam towards the electrode in the microscale, and owing to an intrinsic de-amplification mechanism, this side beam is displaced in the nanoscale, so that a mounted carbon nanotube can be stretched in the nanoscale. Okay, so here you see a fabricated device, and here you can see suspended single-wall carbon nanotubes which were grown directly on the device in this area. So we can't really see the stretching of carbon nanotubes, but we can measure it. So you're probably asking yourself, this probably will end up with a funeral, so how is this going to end? You can see the results in my poster. Thank you.
Yoni?
Hi, my name is Yoni, and together with Yoav Blau, who is in the audience, I'm going to talk about the Nano Antenna project. So, as you all know, the leading solar energy harvesting technology is based on the photovoltaic effect. And this is a nice technology, but it has a few limitations. the most effective PV cell out there right now has an efficiency of about 50%, but it's very expensive. On the other hand, cheap cells have less efficiency. In addition, the materials that are inside the cell have toxic legacy when the cell itself decomposes. So there is another way. It's not our original idea, but we keep working on it. It involves an optical antenna for the infrared spectrum and a rectifying device. Let me start with one element. It's the optical antenna. We designed, simulated, and fabricated optical antennas. We believe that this solution can be both efficient and inexpensive. The other element in our device is a rectifying diode in order to use DC power for electronics. Both of these diodes are based either on MIM, metal insulator MIM diodes, or carbon nanotube diodes. And here we present the integrated solution, both the nano antennas and the nano diodes. We hope to see you in our poster, even though it's not numbered. Thank you very much.
Gilad.
Hi, my name is Gilad. I'm from El Khanin's group. And as you probably all know, neurons were considered to be the building block of the brain, as you can see here, and they were considered as the only mechanism of information signaling within the brain. Another type of cell, which are glial cells, consist most of the brain cells, but were for a long time considered to only have supporting roles and nothing more. Recently, in the last 20 years, it was considered and found that glia have more roles in the brain, such as information processing, coding, and storage. The methodology of signal processing in the brain used to be relied only on the information between the two neurons. But in the last 20 years, much richer and more complex pathways have been discovered, including pathways between the neurons and the glia, pathways between the glia and back to neurons. Our research is in this pathway between neurons and glia cells. Our experimental setup, we take a cortical tissue of brain and plant it on a multi-electrode array, which allows us to electrically stimulate the neurons, while recording using calcium imaging microscopy single-cell resolution of both neurons and glial cells, as you can see here, the neurons in red and the glial cells in green, and by that understand the information signaling between the cells. This is our main result, which, as you can see here in green, the glial cells traces, the fluorescent traces as a function of time, and the neuron cells. In different frequency stimulations, you can see that when you stimulate the neurons in lower frequency stimulations, the glial cells show very little response, while when you stimulate them in higher frequency area, the glial cells show much stronger responses. And this is actually a pretty new pathway of a frequency-dependent neuroglial communication pathway, which will allow us to better understand the information processing in the brain in general. And my poster is 14. Thank you very much.
Ido?
Hi, my name is Ido Amit. I'm from Yossi Rosenbach's groups in engineering. A tunneling field effect transistor is a gated PIN diode that operates in reverse bias under the bend-to-bend tunneling model. You can see here a cleverly rotating tunneling field effect transistor. This is a newly devised system. And I wanted to know have you ever wanted to fabricate your very own tunneling FET? Have you ever wondered which doping method would suit your needs the most? Is the more intuitive VLS method the right method of choice for you, or will you need to resort to the more versatile monolayer contact doping method? Will VLS doping suffice even though it produces a non-uniform axial doping profile, or is the reservoir effect the silent killer of abrupt junction formation? We have abundant data that will help you tailor your PIN junctions to your everyday needs. And we also have some simplified models describing the dependence of transconductance on barrier doping, on barrier tunneling depth. Come join us at poster number 16.
Thank you. Vadim?
Hello, my name is Vadim Krivitsky. I'm from Patolsky Group, and I would like to present you a work, actually a tool that maybe not in the far future will help medical staff to decide which is the best treatment for their cancer patients. Okay, so we're interested in detection of metabolites and oxygen reactive species. Oh, it's better. Oxygen reactive species. So we design multiplex sensing systems for a variety of metabolites. This is how our sensing systems looks. We have here we have a chemical modification that allow us to sense a lot of metabolites that conjugate to oxidation reactions with their oxidase enzymes and measure simultaneously a lot of metabolites. Measuring metabolites and stress, it's really help medical staff to estimate the efficiency of the chemotherapeutical treatment. So here is some results. This is we can see that we have very high sensitivity for peroxides, and this modification also allows us to sense pH. So it's also important when we try to analyze cancer metabolism. Also, here we have some data that shows that our sensing system, sensing method is correlates with a conventional method for detection of ROS, and it's correlates with them pretty good. And here we present of some metabolite sensing like glucose and pyruvate. That's it. Thank you. Poster number 23.
Okay, I'm Lilach. I'm from Hanin Group, and we study quantum dots, carbon nanotubes, photoresponsive surfaces for retinal stimulation. So the retina is a neuronal tissue located in the back of the eye. It's composed of layers of neurons with different functionalities. The photoreceptors sense light and transduce it into an electrical signal, which goes through initial processing by the other neurons in the retina. The output signal goes through the ganglion cells to the optic nerve and to the brain. Blindness can be caused by degeneration of the photoreceptors layer, which means losing the ability to sense light. So retinal implants, which aim to restore loss of vision due to photoreceptors degeneration, are based on light sensing combined with an electrode array to electrically stimulate the remaining neurons in the retina. The state-of-the-art devices include mainly two approaches. One is an external camera, which is wired to an electrode array which is implanted in the retina. And the second approach is an array of photodiodes and electrodes, and the whole device is implanted in the retina. These current technologies still suffer from limitations in spatial resolution, biocompatibility, and light sensitivity. So our approach to overcome these limitations is by creating these carbon nanotubes quantum dots photoresponsive surfaces, where the quantum dots, which are semiconducting nanoparticles, provide the light-induced charge separation. The carbon nanotubes provide good electrical coupling to the neurons, and the flexible substrate further improves the biocompatibility of the whole device. So we developed an efficient method to directly conjugate quantum dots onto high-density carbon nanotube surfaces, which is based on plasma polymerization of acrylic acid. This polymer layer serves as a platform for covalent conjugation of amine functionalized quantum dots. We tested different types of quantum dots, surface coating, and deposition parameters, and we demonstrate the effectiveness of our method by photocurrent and photovoltage measurements, a characterization of the quantum dot loading, and photoluminescence of these carbon nanotubes quantum dot conjugates. And we further performed in vitro tests transferring these conjugates onto mouse retina and recording the electrical activity, and in vivo implantation of carbon nanotubes on a flexible substrate. We are at poster number 36. Thanks.
Hello, my name is Karen Goldstein. I'm working with Emmanuel Peled and the Anna Golomitsky group. My research is about silicon multi-wall carbon nanotubes anodes for high energy density lithium-ion batteries. So the main objective of the research is the development of low-cost, high-capacity, long cycle life, and safer anode material to replace the graphite anode of the common lithium-ion batteries. So the method is the attachment of silicon nanoparticles to the carbon nanotubes. Was developed with the purpose of creating a silicon anode supported by a strong, rigid, and high electrically conducting matrix. The method was prepared by a pyrolysis process, and as you can see in the image, you can see the silicon particles in a multi-wall carbon nanotubes matrix covered with amorphous carbon. So we prepared a characterization of the composite silicon multi-wall carbon nanotubes powder after a pyrolysis process, and you can see here an Ecm image of the silicon covered with carbon and inside the multi-wall carbon nanotubes matrix. More analysis was on the right image. You can see a TEM and IL stem, and we can see the small and nano layer of carbon on the silicon particle, and we can see analysis of XPS, the Bose image on the left, and we can see that after 20 minutes of sputtering of the XPS, silicon carbon can see that formed after the process, the pyrolysis process. And one more technique that we analyzed is an XRD image. On the green graph, you can see the peaks related to silicon particles, crystalline particles, and to multi-wall carbon nanotubes, and to amorphous carbon. After we characterized the powder, we made the electrochemical performance of the composite silicon multi-wall carbon nanotubes anodes to see what is its capacity. And we can see that the composite anode containing not only but 42 silicon under loading of 1.5 milligram per SMR exhibit large capacity of 2040 milliampere per gram, and capacity after 100 cycles performed 835 milliampere per gram. This is more than 2.5 times that of conventional carbon. And thank you all for attention.
Hello. First of all, let me thank you for the opportunity to present my work here today. My name is Turchinsky Dmitry. I'm from Yuval Ebenstein's lab, and we deal with epigenetic modifications of DNA molecules, and we study them by single molecule fluorescent microscopy. My project is to develop an assay for quantitative measurement of telomere length distribution. So what do? I take a DNA sample, I introduce a reference molecule synthesized and pre-labeled by PCR. Then I dye the mixture with intercalating dye, picture the sample in two channels, and I do an overlap of the two images to understand which are the reference molecules. The next step is to extract data. So I build a program in CVI which extracts data from the shapes. Then we do statistical analysis of the extracted data to see the distribution of the shape intensities. Here is the number of counts. We see here an example of 82 pictures of a sample containing 3 and 12 kb DNA molecules. 82 pictures, 8,000 shape extracted. We see here two populations. Here we did fitting on different bin sizes. We see the phenomena is repetitive on different bin sizes. It's not something artificial. In the future, we will have a reference peak here. By knowing the reference peak, we can characterize the other peaks. So if you want to know what telomeres are, how they're connected to my work, and how I'm going to measure them, come and see my poster. And thank you very much for your attention.
Okay, Omer, I think the last one.
Hi, I'm Omer from Chesnovsky Group, and our challenge is to develop label-free super resolution. Background. The diffraction limit is the physical limit of resolution and defined as half of the wavelength. For the last year, several techniques in fluorescence microscopy demonstrated sub-diffraction limit resolution. One of them is TED. In this technique, a donut-shaped beam called a STED beam creates a small effective area of fluorescence emission by depleting the fluorescence molecule and putting them in a dark state. This scanning method reached resolution of 20 nanometers, but with fluorescence dye. Our goal in this research is to develop label-free super resolution without fluorescence dye by using Raman spectroscopy. And how does it work? An ultra-short laser pulse excites the sample and creates transient thermal effect. As you can see in this simulation, the pulse heats up at the sample and creates an instantaneous temperature gradient inside the diffraction limited spot. Each part of the sample with different temperature affects the Raman spectrum a bit differently. And how? Look at the Raman spectrum of silicon. This is in room temperature, and as it gets hotter, the Raman peak shifts and broadens. So, what we do, we impose a spectral region of interest on the Raman spectrum, and this region of interest enables us to get here, in the cold tail, a small effective area inside the donut shape. So using the heating pump and the Raman probe, we raster scan the sample, and look what we get. This is a standard resolution test in which two Raman point sources are located 120 nanometers from each other. And now we scan the sample. First, you see here, with the diffraction limited Gaussian pulse, we scan the sample and you see that the two targets cannot be detected. With our method, we can detect the two targets. And this gives us an improvement of a factor of 3 to diffraction limit. And I remind you, it's label free. So I invite you all to understand more in poster 27.
Okay, so let's thank all the speakers.